So hello again, everyone. Thank you for coming on our today for our webinar on the neuromuscular junction in vitro model and also the ALS applications. So today for our today's webinar, I'm joined by two of my colleagues, Adriana Thomas-Huel, who is our product owner in the biopharma applications. She has a background in microfluidics and also cellular biology. And we also have in remote Jessica Ronta, who's our scientist also in the biopharma domain. And she has a lot of experience in CNS and also organoids. So just to give you a quick overview of our today's program, we'll of course show you how we were able to characterize iPSC-derived human motor neurons, both in healthy but also in patient donors, and also the myotube characterization. And then in the end, we'll show you the neuromuscular junction model using our microfluidic devices. And as usual, we'll end our session with a Q&A part where you can ask your questions. Of course, if at any time during the presentation you have any questions, do not hesitate to write them down in the chat and we'll get back to you in the end. So just to give you a quick just a quick overview of what we do at NetTree. As we all know, when we want to study complex systems or we want to do screening, drug screening, we usually will study mixed cell types using 96 or even 24 well plates, where we usually use mixed cell types such as neurons and, for example, target cells. But we know that there's a natural compartmentalization in the human body. The first one, the compartmentalization between the external stimuli, the exterior environment, and also the internal, but also between different types of organs and even between the PNS and the CNS systems. And so what we do in our devices is that we recreate this physiological compartmentalization. As you can see on the right, you have the architecture of our dual link chip, where you can see you have 3 channels with 2 main channels on the left and on the right. And in between you have a second channel that mainly acts as a microfluidic barrier, which means that the liquids from channel 1 and channel 3 are not in contact. And this helps us to actually in a way that when we will seed, for example, sensor neurons, as in the schematic in the middle of the screen, you can see that by seeding sensor neurons, for example, in channel 3, at the end of the culture, you'll have cell bodies that stays in the channel 3. And then you only have axons that go through all 3 channels, and then they can innervate your target cell, whatever cell types you seed in the first channel, for example. And in this way, you have you can recreate the natural compartmentalization where cell bodies or somas, they're not in contact directly with, the example, if you want to have a model of skin model, for example, your somas will not be in contact directly with the keratinocytes, and or, for example, pulmonary cells. And the only ones that will be in contact with your target cell will be only the nervous extremities. So this is our most standard and classic device for 2D cultures. We also have created recently in early access a 3D culture device, which is the one that you can see down below. It's a device where we have a middle well, where you can culture organoids or different mixed organoids. And you have a bottom reservoir where you can see, for example, endothelial cell to recreate a vascular system, which will be separated from the central well by a porous membrane. And that way you only have the exchange of ions or different types of molecules. And so these, at Netry, our chips are made from, as you can see, they're transparent. So we make our chips from PDMS, which is a type of silicon that is, first of all, is permeable to oxygen. So you don't need extra pumps or other devices to maintain your cultures. Because the chips come in a plastic frame that has the same size as a 96 well plate, it can be compatible with most lab equipment. And in this case, in this slide, you can see our the MEA device, where you can see it on the image. You have underneath the microfluidic channels, you have microelectrodes that can capture the electrophysiological signals of neurons. And in this way, we can see that neurons as biodigital sensors. As we know, they interconnect different organs, or they can transfer different messages coming from their environment, whether it's heat or cold, and even vehiculate between different organs. And so what we propose today is to actually use this MEA technology to capture electrophysiological signals of neurons and their interconnection. It's what we call digital signatures, which is how neurons react to a certain stimuli or a compound that we want to screen, in comparison to reference compounds that we know they have a specific efficacy or a toxicity. And in that way, we're able to have predictive models. And so, of course, we do sell directly our devices to our clients, but we have our own R&D labs in biology, where we develop different types of models. And so we also propose screening services and also co-development to create new models that we don't have in our lab yet. And we're trying to target different types of application, going from oncology, immunology to cosmetics and gut. Of course, for the screening services, we can study at different levels, from the less complex ones, such as studying compounds or how they affect the response of neurons only. But we also can complexify the model by using cell supernatants on the neurons. And then, of course, we have the most complex one, the co-culture. And so I will let Adriana take the lead and discuss the neuromuscular junction model.
Thank you very much, Nasiba. We're not going to talk about our NMG platform, and particularly its application in motor neuron diseases, focusing on the C9 or F72 mutation involved in genetic forms of ILS. We have combined two innovative technologies, the use of differentiated cells from iPSCs and our microfluidic technology, to recreate an optimal cellular microenvironment and test different readouts. I would just like to stress first that these data that we'll further present have been produced thanks to our partner, Axol, supplier of iPSC cells. And prior to the co-culture, we're not going to take a look at how the cells look like in a monoculture. So the first cell line to be characterized is motor neurons from apparently healthy donors, age 74 years old. In these immunofluorescence images, you can see the expression of key markers, characteristics of motor neurons, in particular, choline acetyltransferase, CHAP, an enzyme that is responsible for biosynthesis of neurotransmitter acetylcholine, and HB9, a transcription factor described as a spinal cord motor neuron specific marker. We can also see to the right of the image electrophysiological data that have also been obtained, highlighting the presence of synchronous bursts and constant amplitude spikes. The same characterization has been done for motor neurons, but this time from patient, a 64-year-old woman, that carried the C9 or F72 mutation. We can observe the presence of markers such as CHAT and HB9 in the fluorescence pictures, but we observe a different morphology and a disorganization in vitro with hyperexcitability and spikes of variable amplitude compared to the healthy line. So there are two different cell lines currently available for motor neurons, so cells from healthy donors, and another one from a patient carrying a genetic mutation. So in vitro, these two lines show a distinct phenotype in terms of expression of phenotypic and electrophysiological markers. Next, I will show you data on monoculture for the myotube differentiated from iPS. So immunofluorescence staining was performed, highlighting the expression of key markers such as myosin, a major component of skeletal muscles, and titin, a muscle cell protein, as early as 5 days in culture. These further images also highlight the presence of multinucleated cells, which are very characteristic of muscle cells. Here, you have different representations. Muscle cells from healthy donor express key markers such as actin and dystrophin and have the expected sarcomere structure that you can see here highlighted in yellow to your left-hand side in the bottom. So once the two cell types have been characterized to ensure a full differentiation process and the presence of key markers, co-cultures were performed in our microfluidic devices that Nasiba had just presented in the beginning of this webinar. For these preliminary experiments, we have used dual-link devices. These devices are composed of 3 compartments, 2 channels, 1 and 3, for cell seeding, and a second central compartment, channel 2, for fluidic acylation, as Nasiba very well explained it in the beginning. Each channel is connected by microchannels through which axons only can pass. The device may or may not be coupled to electrodes to make electrophysiological recordings over time. So in concrete terms, muscle cells were seeded in channel 1 and motor neurons in channel 3. Initial results show that the co-culture is viable in our microfluidic device for more than 20 days, making it possible to monitor electrophysiological recordings over time, with motor neuron extensions entering to the muscle compartment as early as day 10. And these fluorescence images, in these images, actually, you can see the artificially colored red extensions of the motor neurons going to the muscle compartment, artificially colored here in green. An experiment was also carried out with alpha bungarotoxin, shown in white, in the image on the right, which is extracted from snake venom and binds with high affinity to the alpha subunit of the nicotinic acetylcholine receptor in neuromuscular junctions. So this underlies the functionality of the neuromuscular junction. So in conclusion, we have a platform for co-culturing muscle cells with motor neurons, enabling electrophysiological recording of the activity over time, immune labeling that can also be carried out, and various compounds involved in motor neuron disorders that could be tested, or bottle-like compounds to be investigated for desmoid cosmetic applications. Please, Nasiba, you can take it over from here. Thank you.
Thank you, Adriana. Hopefully everything was clear and you have questions if you want to learn more. Of course, in the end, don't hesitate to contact us if we didn't have the time to respond and reply to everyone. I think Jessica can also join us for the Q&A session. So let's start to look at the questions. So the first one is, hello, thanks. The CME data is after 10 days after plating. Yes. In some of the slides, in fact, it was 10 days after plating, but we also went up to 15 days after plating too, in the co-culture model. Do we have other questions? In your platform, is it possible to add compounds to induce pathology without having access to cells from pathological donors? I think, Jessica, you're the best one.
Sure. Yes, absolutely. It's possible. Thanks to fluidic isolation, you can add a compound to channel 1 or 2 or 3, such as, for example, TDP43 oligomers to induce a pathological model.
Okay, hopefully this was clear to you all. This one is more for you, I think, Adriana. In your platform, would it be possible to follow the rapid electrophysiological activity of a compound for less than 5 minutes?
Yeah, of course. I mean, you can use your compound and you can measure the baseline of the neuronal activity and then add your compound in the axon collecting system, and you can follow immediately the response of your compound and how the cells respond to the compound. So it's less than 5 minutes. You can also, of course, measure the activity over several periods of time, since electrophysiology is not an invasive method of measuring.
Okay. Some questions on the muscle cells. Do the muscle contract spontaneously? Do you have detachment of myotubes? Can you help us with that, Jessica?
Yes, for the moment, it's the preliminary data. We don't observe muscle detachment, but we observe some muscle contraction, but it's preliminary data. We have to repeat.
And so do the NMJ show an ALS phenotype?
Well, I can maybe respond to that.
Yep.
I mean, the neuronal cells had the phenotype, but not the muscle cells. So in this co-culture, only the neurons, the motor neurons derived from the patient, carried the mutation, C9 or ERF, but not the muscle cells were healthy, from a healthy donor.
Okay. Another question, probably for Jessica. Would it be possible to use your platform to monitor abnormal protein aggregation?
Yes, we didn't do this in the study, but yes, it's possible. As you have seen, our devices are compatible with confocal macroscopy, so no problem to see that.
Okay. Another question on myocytes. In this platform, is it possible to generate bundles on myocytes that can be excited for movement? I don't know, maybe Jessica, what do you think?
Yeah, say that Jessica should answer.
To generate bundles on myocytes. To be honest, we don't have checked this, but next step, I think.
And what about the readouts on the muscular compartment? We talked a lot about motor neurons, I guess. And can we look at the readouts on muscular compartment and look into muscle degenerescence, related diseases or mutations?
Yes, we can use the same readout for the neuronal compartment, can be used for the muscle cells, such as immunofluorescence, morphology observation, but also the release of acetylcholine directly into the supernatant can be evaluated also.
Were modern neurons and muscle cells seated at different times in the microfluidic device? Maybe Adriano Or
Yeah, sure. So the two cell types proceeded at the same time for the co-culture. So they evolved together until the co-culture was reached. And as I've mentioned, the neuronal projections could already be seen at day 10 in culture. We have not tried different, our partner hasn't tried different timings of seeding.
okay. Thank you, wonderful model. Can you adapt the microfluidic plates to MA Maestro? Is that the MA used to evaluate ALS phenotypes?
Yeah, I can answer this question. Yes, indeed. We did use the Maestro system, and our plates are compatible with the system, and it was used to evaluate the electrophysiological data from the neurons.
Okay, thank you. Okay, we have another one. Is it possible to make the platform more complex by adding other cells? Jessica?
Yes, we are presenting here motor neurons with muscle cells, but you could imagine motor neurons with cardiac cells, for example, of course, with cell culture optimization, or we can perform a free culture with the addition of glial cells such as astrocytes to increase neuroprotection, for example, of course.
And would it be possible to monitor axonal degeneration of motor neurons in your platform?
I can take this one. Yes, thank you for this question. Yes, absolutely. Different options. First, for example, using GFP cells, for example, or by adding a non-invasive fluorescent compound for live cell imaging, or by fixing the cell at the end of the experiment and performing immunostaining and quantify, for example, a maximum length or a number of axon fragments. Please feel free to contact us for more information on this topic, because we have extensive data on motor and sensory neuron regrowth after injury in the axonal degeneration context.
Thank you. And did you test different cell densities in the microfluidic devices?
Yeah, as I mentioned earlier, we haven't tested so far different seeding times and different cell densities, so I think it's to be potentially tested in the future. These are preliminary results that are showing here.
Okay. And possibly for you again, Adriana, is it possible to record neuronal activity several times on your platform?
Yeah, also, I think I sort of responded earlier to that. The MEA technology is not invasively, so you can record over several periods of time within the same days and several days. So as long as your cells are viable, you can record the electrophysiological activity.
Okay. Okay, we have another interesting question. Would it be possible to use botulinum toxin A or butterfly compounds in your platform?
Yes, yes, there is no problem in adding compounds to macrofluidic channels and, for example, recording electrophysiological activity after. However, there are some regulatory requirements associated with the use of toxins in France. So please do not hesitate to contact us directly on this specific point to evaluate together the quantity of toxin you need, but of course it's possible.
Okay. How many NMJs are seen on average in one chamber, and how long can NMJs stay alive?
So up to now, I mean, how many NMJ we haven't quantified, but we see, I mean, from the images that I've also shown a bit earlier, is that we see different spots where the where the toxin is present and representing the neuromuscular junction. We didn't quantify that. And regarding how long the NMJ stays alive, I think in comb culture, correct me if I'm wrong, Jessica, we can be more than 20 days, but we still need to see if we can push it further than that.
Okay, do we have other questions? Okay, so I think we have replied to a lot of your questions, most of them. I think that's it. Do not hesitate if you have other questions coming after the webinar, things that you think about. Don't hesitate to recontact us. Okay. Yeah, don't hesitate to recontact us on contact@netri.com. We'll be more than happy to reply to those questions. Do not hesitate. If you're interested in our devices, you can look at our website. And thank you, everyone. Hopefully this was really interesting for you and you've learned a lot. And yeah, thank you, everyone.
Thank you. Goodbye.
Thank you.